Good evening, everyone. Today I will be discussing about lactic acid bacteria. Okay, how the lactic acid bacteria is lactic acid bacteria. What are the uses? What is the application? Okay, so we will be discussing about lactic acid bacteria here. Now, the basic points what we will be talking about is what is the basic things, and this particular lecture has been specified mainly, it is under the fermentation technology, and the lecture is specifically meant for the UG students and the diploma students of food process engineering and food technology, biochemical engineering. Okay, so we will be basically, why I took the topic is like sometimes there are some questions you will often get in competitive exams also. So based on that, like few insights I would like to say about lactic acid bacteria here. You can refer this as a outline note of yours, where I'll just throw the key words and the key points here. So let's start with it. The first thing what I will be talking about for lactic acid bacteria, as we know, that is CH3CH, CH3CH, OH, COOH. Okay, so CH3CH, OH, COOH. This can be the. This is what is the I have taken. I am writing it as the formula of lactic acid bacteria. Now, what you can tell is you will be usually how you are getting the lactic acid. Mainly, they will be following the solid state fermentation. Okay, the solid state they will follow, as well as how does the lactic acid looks. The lactic acid usually will be in the solid state. It is usually water soluble. Okay, the characteristics. Usually it is solid state. It is water soluble, and you are mainly using this. Mainly you will be getting this in the source of lactic acid you are aware of, right? It is mainly for the dairy products. Then mainly you will be getting it in cheese, yogurt. Okay, cheese, yogurt, kefir. Yes. So in these products, you will be often getting this lactic acid bacteria as a source. Mainly the characteristics will be solid state, water soluble, as well as you will be having it will be definitely lactic acid acts as a preservative. Okay, this is another part. It acts as a preservative. Okay, so this is the first thing. Then the next thing is about, we have the embedded Meyer-Hoff-Parnas pathway that you are aware of, the EMP pathway, where a part of this we have you might have studied in class 2 in biochemistry or biology, where you have studied about the embedded Meyer-Hoff-Parnas pathway, where we have seen the glucose is getting converted into pyruvic acid. Okay, this part, and from there we are in the presence of this NADH, NADH. Okay. So NADH will be giving you the NAD plus. In that NAD plus, and we will be getting the lactic acid. Okay, this is how we are getting in. Naturally, it is like embedded Meyer-Hoff-Parnas pathway. We are getting this. In the glucose will be first there, and this glucose will be converting into pyruvic acid, from where this NADH will release, and that NADA plus, and you will be, it is getting reduced to NADA plus, and this NADA, and as a result, you will be the lactic acid formation will take. This is one part of the about lactic acid. How do you get it? So what we have discussed now, just quick like points we can say. It is a solid state. It is in the solid state. It is water soluble. It acts as a preservative. Lactic acid will be usually from you will get from the embedded Meyer-Hoff-Parnas pathway, where we will be EMP pathway, where from the glucose it will come to the pyruvic acid, and from there you will be getting the lactic acid. Okay, now the next part which we will be discussing is, we will talk about the lactic acid calcium lactate. Now we can say what is, see, lactic acid or calcium lactate. If we talk about the polylactic acid, if we are talking about polylactic acid, polylactic acid is like, it acts as an adhesive. It will act as an adhesive, polylactic acid. Similarly, if I'm talking about the lactic, see, L-lactic acid and D-lactic acid, there are two types. Like you have L-glucose and D-glucose. Similarly, we have the lactic acid in two forms. One is L-lactic acid, another one is D-lactic acid. Okay. Now, what are the characteristics? So we have L form and D form. L-lactic acid, D-lactic acid, two types. Polylactic acid is usually used as an adhesive. This is one point you can take. Now, what we will talk about is what are the lab characteristics. Lactic acid bacteria is also called as lab. And what are the characteristics of that? It is diverse in nature. You will be getting that in diverse. It is diverse. That means we can try and find it. Like it is not in scarce. Okay. Then one thing is it is a gram-positive bacteria and it is non-sporulating. So what you understand by, so why lactic acid bacteria? This is gram-positive. This is one point you can take. And it is non-sporulating. Okay. Ability to produce lactic acid. The lab bacteria, lactic acid bacteria will be producing the lactic acid surely. Hence the name lab, lactic acid bacteria. It is anaerobic. This characteristics, please remember, it is important. Sometimes it comes like, in MCQ. The only gram-positive, the principal gram-positive bacteria present in dairy products, you may know, that is lactic acid bacteria. A non-sporulating gram-positive bacteria, you can give an example, lactic acid bacteria. Okay, and anaerobic gram-positive bacteria, lactic acid bacteria. So, okay, these points, please remember, this can be a MCQ type questions. Then it is like tolerate the aerobic. It can tolerate aerobic and the pH of is around 3 to 6, but mainly it is anaerobic. Okay, high ethanol concentration, it can withstand microaerophilic, and the main uses which we can use in that, you will be having in medicine. We use it in medicine, lactic main uses medicine. Second is in the food. Okay. Now, cucumber and cabbage, if you say, cucumber and cabbage, we are using the lactic acid bacteria. Okay. And the lactic acid bacteria will have a group which will be telling, that is lactobacillus, we can say pediococcus, okay, leuconostoc. So these type of bacteria, they will be helpful. If you have heard about sauerkraut you have, that is the cabbage which is getting shredded. It is formed, it is like this microorganism plays an important role in sauerkraut production. Sauerkraut is mainly of cabbage. So if you are not able to like, what you can say, if you are not getting the particular one of these species, then we cannot follow. Matlab, the sauerkraut production will be hampered or it will not be in the correct way. You will not get the sauerkraut product. Okay. So if I come back and conclude here about this particular slide, I can say lactic acid is formed in two forms. That is your L-lactic acid and D-lactic acid forms we have. Then the second thing is the lab is diversification. That is lactic acid form is in diversification, or it is diverse in nature. The point which you should know is it is having gram-positive bacteria. So if it is gram-positive bacteria, this is gram-positive bacteria, non-sporulating. Okay, then the pH is 3.6. This is important. Then there are some bacteria like lactobacillus, pediococcus, leuconostoc, which are important in fermenting products. One such example is sauerkraut. So next we will be talking about the What do you understand by homofermentative and heterofermentative? There will be one difference, right? And obviously the lactic acid is having some antimicrobial properties you are aware of, must be. That is it is acting as a biopreservative. Okay, so the points are very simple. Just if you act from this lecture, if you just take the small points, it will may be useful for your sometime in an interview or some questions you are getting in your exams, that might be helpful for you there. That is the idea behind. Okay, then you can tell about what you understand by homofermentative and heterofermentative. This is important. Homo means what? Homo means homofermentative. Homofermentative. And another one is heterofermentative. What do you understand by homofermentative and heterofermentative? Homofermentative is like kind of, you can say, I have discussed just now I said about embedded near Horner's pathway. Only the lactic acid will be formed. Okay. One such example is Lactobacillus casei is a homofermentative. It comes under this homofermentative lactic acid bacteria, as well as Lactobacillus vulgaricus. Lactobacillus casei is sometimes used for curd as one of the culture. Lactobacillus vulgaricus we will use along with Bacillus thermophilus, Bacillus thermophilus and Lactobacillus vulgaricus. Both together combinedly, we will go for this yogurt formation. Streptococcus thermophilus. Okay, I'm sorry. It's Streptococcus thermophilus and Lactobacillus vulgaricus. We are using for yogurt. L. casei we sometimes use as a culture for curd. For when we talk about the heterofermentative, one such example is Lactobacillus acidophilus. And here we will get, like the pentose pathway, we know, pentose phosphate pathway, we will get where lactic acid will not be the only end product. There will be some other along with lactic acid. Lactic acid will not be the main. So it will be a combination of like it is heterofermentative and homofermentative. There is a differences. You will be getting only lactic acid, but for this purpose, we won't be getting only lactic acid, but some other products also, or from some other components you will be getting. So one such example we have is that is heterofermentative, that is Lactobacillus acidophilus. Okay. Then we have what is the importance of this lab? There should be some importance, right? Why? Why do we need lab? That is Lactobacillus lactic acid bacteria we need. Why do we need that? What is the importance of that? The importance is like we can use, I'll just count. The importance over here is we will use this like importance. What is the importance? The importance is like if yeast will utilize the carbon source. Yeast utilizes carbon source. Okay, the first point. Second point is it is acting as a preservative. As well as avoiding the putrefying bacteria and prevent, it avoids the putrefying bacteria and prevents contamination. Okay, the spoilage reaction, it is responsible. This is the first beneficial part you can take. What is the other aspect of lactic acid bacteria? It causes turbidity, flavor loss in beer and wine. Acetone butanol fermentation it leads. Acetone butanol fermentation. Okay. Then one more is that leuconostoc mesenteroids. They will interfere with the sugar crystallization. They interfere with sugar crystallization. For meat also, sometimes the surface growth leads to the spoilage. Here, what I did was, I talked about this lactic acid part regarding what are the basic problems which we are having in the importance and spoilage. You can take this part, like I have mentioned here, equinostalk mesenteroids is creating some sugar crystallization problem, interferes, then needs surface spoilage. So take some examples of that, and you can read in detail that what are the spoilage microorganisms regarding to lab that is not beneficial. Similarly, the beneficial microorganisms also you can take. So here, what the few takeaway points again we can say is, what are the typical microorganisms we can have which is beneficial, the lactic acid bacteria beneficial, as well as which is leading to indicating spoilage. These are the things. Now, I will be taking one simple example and I will tell you how you will be getting this particular lactic acid formation. Okay, how you will get the lactic acid from whey. So this is one production step. Let's go with it and see how we can get this is your whey, how you will get lactic acid from whey. Okay. So whey at 40 to 43 degrees Celsius, this is the time, and 42 hours is the temperature. So time temperature combination of whey, this will be taken. Now we are taking the calcium hydroxide solution in the form of a slurry. Okay, that should be less than 0.1%, less than equal to 0.1%, which we are taking. Okay, now this calcium hydroxide along with calcium hydroxide in the form of slurry we are taking. Now this calcium hydroxide, what we are taking, this along with lactic acid will be giving you calcium lactate. Okay. So the advantage is that it will take shorter fermentation time. Fermentation time will be short, as well as you will get higher yield. Yield is high. Okay, that means what? We are taking the whey. We are taking the whey and this calcium hydroxide we are taking in the slurry, and we are adding that. Along with the lactic acid, which we are getting in the calcium lactate form. Okay. Now, we'll be having one point here. This is one. Another point we can talk about. Another point what we can say is, if you take cheese whey, we are taking cheese whey, we need to doing ultrafiltration. We are getting whey protein. Okay. Now the ultrafiltration we are getting whey protein, then we are going for fermentation process. By how? We are inoculating with lactic acid, lab. Okay, then we'll go for nanofiltration. Around 2 to 10% lactic acid we are getting here. Then we are going for the reverse osmosis process, where we are getting the lactic acid alone. Go for vacuum evaporation. 90 to 98% lactic acid we are getting. Next, we will go for polymerization. If we go for polymerization, we are getting polylactate. Okay. Next, this is the from cheese way. If you have, you will go for the membrane separation technique. This is one way. Okay, we are doing inoculation with LAB, ferment it. Then nanofiltration, we go ahead. We will get 2 to 10% of lactic acid, which on reverse osmosis, we will get the lactic acid alone. That if you evaporate out, you will get 90 to 98% of the lactic acid. Okay, that when you go for polymerization, it is giving you the polyactide. So here, what are the unit operations involved? We need ultrafiltration, we need nanofiltration, we are going for fermentation, reverse osmosis, and then on with vacuum evaporation, finally the polymerization. So then what we do is, if we have, this is how we will get the lactic acid, polylactide from whey, cheese whey. The next point what we are getting is, how will you recover this? Because we have already got that. Now, what we will do is, for recovery process, till now we are up to polylactide. Okay, we took the cheese whey, we went for ultrafiltration technique. From ultrafiltration, we got the whey protein. We went for nanofiltration, we gave the inoculum. From there, we are getting around 2 to 10% of the lactic acid. From 2 to 10% of lactic acid, reverse osmosis, we are getting lactic acid. Vacuum evaporation, if you are doing, you are concentrating the lactic acid alone with 90 to 98% yield. That will be polymerized and you will get polylactide. Now, how will you recover the process? When you are recovering the process, it is like calcium hydroxide we need to add to the broth. Calcium hydroxide is added to the broth and that is heated. We are adding this calcium hydroxide. Okay, initially I have discussed with that. I have told you what we are doing it. So we are adding this calcium hydroxide, heated at 80 to 95 degrees Celsius. Okay, and then we are getting the filtrate. This filtrate, whatever we are obtaining, that needs to be evaporated. That will be evaporated out. The calcium lactate we will be getting. This calcium lactate we will dissolve in water. Okay. Decolorize it with the help of filter. For around 15 minutes. It is a filter for 15 minutes. Then the calcium lactate we will get will be with dilute H2SO4. Clear? Now this is how we can recover. Dilute H2SO4. Then we will get the calcium sulfate and lactic acid. Calcium sulfate will be precipitated out and you will get the lactic acid. This lactic acid which you are getting, okay, that will be the fermentation product. So what is the ferment? So idea of sticking all this recovery part is nothing but they will not, you have to know what are the, where you are getting, what are the products we are obtaining. So mainly we are obtaining the lactic acid. But the lactic acid which we have obtained, that is nothing but the calcium sulfate is precipitated out and lactic acid is formed. Now this calcium lactate along with the lactic H2SO4 in the reactor we will be sending it in the reactor. Okay. So calcium lactate, whatever you are having, calcium lactate, we are either going for the H2SO4 where lactic acid we are getting, or whichever calcium lactate we can do two ways. One is the dilute H2SO4 and getting this calcium sulfate and lactic acid, or the calcium lactate can be kept in a reactor and centrifuged. Reactor at 30 degrees Celsius we will keep, and this we will centrifuge. Okay, we will centrifuge at 80 to 95 degrees Celsius, where we will get calcium sulfate that will evaporate out. In filter you will get and filter. So what is the product we are getting? Crude lactic acid. I'll just remove it and tell you again. Has become much. So just from here, I'll tell you again what is happening. We are getting the calcium lactate. Okay, the calcium lactate needs to react with dilute H2SO4. So when we are setting it in a reactor, okay, at 30 degrees Celsius, right? Then we will centrifuge this. After mixing, we will centrifuge it at 80 to 90 degrees Celsius. After centrifuging, we will be getting, we need to evaporate out. Evaporate and filter it. So you will get the crude lactic acid. Okay, so this is how we can get it from the cheese way. I have given you the flow sheet, how you can get. So this crude lactic acid need to be purified again for edible purposes. Okay. Right. Then one more thing is, we have different, so we have got this crude one. Okay. So if I am talking about the crude lactic acid, and if I'm talking about this one is the crude lactic acid which we get. This one, or you can say about the technical one, which is having around 20 to 80% purity. Further, we need to purify the process. So we will get the edible one. Okay. When we get the edible one, it is around 50 to 80%. We'll have 50 to 80%. And when you are having the pharmaceutical part, the plastic part will be used, usually 50 to 80 again when you are using a polylactide, polylactate, and when you are using for pharmacy or pharmaceutical aspect or medicinal purpose, it should have a much purity of greater than equal to 85%. Okay, so crude lactic acid usually you will use in the textile industry and all. Yes, for delining, for textile industry, as well as for edible lactic acid. Where do you use? We can use in the casein. We can use in the casein precipitation. Even sometimes, so we will use in fat extraction, casein precipitation. So these are the uses. So with this, I would like to conclude this lactic acid lecture session. Just the key points what you need to take that I will write in the, what are the key points you have to take. So the key points. lactic acid bacteria, what type it is. It is anaerobic. It can withstand aerobic, but non-sporophilic and anaerobic. This is one thing. What type characteristics of lab? Then you can get lactic acid naturally from the EMP pathway and phosphate pentose pathway. One such example. Then you can even get lactic acid from the cheese way, or even way. One such I have given. The different types or the grades of lactic acid. That is the crude, edible, technical, crude or technical, edible. Then it is used for pharmaceutical, like plastic. Okay, what are the grades? Then what is the benefit of lab? Benefit as well as harmful. Lab can be both benefit as well as harmful. So what can be that? Yes. Then besides this, what are the applications of lactic acid? Where all you can use that you can tell about benefits and harmful together. Like you can say it is used for this lactic, and another one is about the source from where you are getting lactic acid. That mainly the dairy products. So these things which are mainly you should focus on, that will serve the purpose of this particular lecture. The lecture is taken by myself, Payal Gupta, research scholar, Department of Agriculture and Food Engineering, IIT Kharagpur. Thank you for listening. I hope it will help serve you some purpose for this lecture. Thank you so much.